Thanks so much. And I've been asked to give a talk today on a novel technology, novel imaging technology that we developed at Emit Imaging. And thanks for your attention, and I hope you're having a great conference. So the title is Block-Based Tomography with Applications in Neuroscience and Research. Just by way of background, my name is Jack Hoppin. I'm a mathematician. I was one of the co-inventors and catalysts for the technology. It's a relevant context to tell you that I founded and was running a company called Invivo, where we developed this technology, and we started Emit Imaging to really fully make an end product of this modality that I'm going to teach you about today. By way of context, while I was at Invivo, I also we were acquired by Konica Minolta. I oversaw Konica Minolta Precision Medicine, and in that role really had the pleasure of overseeing a genomics, pathology, and radiology division. And I want to give some context of how I think that those three disciplines really are a continuous spectrum of studying biology for use in this context of today's presentation, kind of basic science research, and I'm going to be focusing on a neurology application. So I'll motivate this new modality in three-dimensional tissue digital pathology and talk about the Xerra system that Emit Imaging created. And I'll give you a little background to how we got there, talk through the system, and go through multiple applications. And then the lion's share of the presentation will really be around an exciting effort we made in fluorophore and radiolabeled antisense oligonucleotides. And I'm going to present what I feel to be sort of the killer ADME DMPK study for novel therapeutics, in which this technology played kind of the critical bridging role. So this is a modification of a publication we made, kind of talking about the bridged gap between pathology and radiology. Historically, we could think about microscopy as a many-century-old effort when we first started looking at cells under a microscope, and radiology is a century-plus-old discipline. And what's happened over time is, with technology, we've really been able to build a continuous imaging capability between the subcellular environment and the whole human. This is a little bit of a forecaster of some intrathecal work that we'll be showing later. And you see a SPECT CT on the upper right and a fluorescent microscopy image on the bottom left. And that maybe 15 years ago, we see a microliter activity. That's the radiolabeled antisense oligonucleotide spinning mouse in the middle. And that was a breakthrough, and it was really kind of bridging the gap to begin with, and being able to do micro medical imaging in the discovery space. It's been a huge help and guide to do in vivo studies of pharmacokinetics of drugs, of pharmacodynamics of drugs, to look at all sorts of biology in that, and pharmacology in that space, and chemistry. We see there next to it is an image from this modality, which in this case is the fluorescent labeled antisense oligonucleotide. And it's a kind of a nice three orders of magnitude volumetric look. We're kind of a milliliter sport in humans, a microliter sport in rodents. The technology I will talk about is a nanoliter sport, and we were under a microscope. We're even down to femtoliter. Getting carried away with this concept, and you can see we really also could do this kind of across the whole electromagnetic spectrum, and we'll talk about that in a moment. This is a cool image from a technology that Konica Minolta developed called QuantaCell that enables quantitative immunohistochemistry of cells under a microscope using a nanoparticle technology they developed. And I used it to kind of jump to the next page, where that quantification comes from an attoliter electron microscopy image of those nanoparticles, which are the images they use to actually make it a quantitative immunohistochemistry assay. And if we keep on going down the line all the way to a yoctoliter, which is not a unit of measure one typically uses, or probably should use, we see a really cool image from the University of Cambridge, where they're they're really looking at the first three-dimensional structures of active DNA. And I use this as an inroad into what I feel like. I think neuroscience is, well, I really like all these areas of research. Neuroscience is one of the coolest areas of research, one of the most complicated, and it demands, and I think we'll show today, a huge merger of imaging fields. And I put imaging in quotes because a lot of the modalities I'm going to discuss on the slide briefly wouldn't be considered imaging. But in the previous slide, I think I view imaging as any digitization of biology, whether it doesn't need to be a picture to be an image. It can be some quantitative readout or analog readout, frankly, of biology. So from the previous plots, we showed a very large dynamic range. Humans are about 150 liters. And again, not a good unit of measure for genomics, but we set the octoliters down to a single variant. And that's 10 to the 26 dynamic range to interrogate. We can think of the human body as having, I think, 30 trillion cells. And so it makes sense that we need a huge dynamic range to study it. We can image across all disease areas. We have lots of workspaces in which we do research, and I'll comment on kind of the workspace we're in here, a discovery workspace and leading to a first in human space. But we use these technologies across those diseases and those levels of biological interrogation, just about from bench to clinical practice. There's all sorts of chemistry we need to do in imaging, and from a modality perspective, whether it's a high energy gamma ray in PET or a radio wave and magnetic resonance imaging, we cover a 10 to the 13th dynamic range across the electromagnetic spectrum. This enables, to the right, all different types of imaging. I'm going to be focusing a lot on molecular imaging today, and it demands, and the focus of this talk will not be on, I feel like every talk has a talk about artificial intelligence, but we do use, there's actually a lot of deep learning that has gone into some of this work, but there's so much information coming out of these efforts. You just can't ignore how much computational power has gone into these technologies, as well as must be used to dimensionally reduce all the data to some rational decision or endpoint. I highlight now in this next step what I think is relevant in the Society for Neuroscience, and in this case, it's really everything except the disease areas, neurology. But it's such an interdisciplinary activity, and there's a lot of data requiring a lot of expertise. And however powerful someone's intellect may be, I think it's not hard to imagine that it is an interdisciplinary sport with a breadth of expertise needing to come together to be successful. And so there's many people that have contributed to this work, as well as, probably 7 or 8 disciplines that came together to show you the oligonucleotide study. That'll be the highlight. I think academic and pharmaceutical programs are really the best launching ground for these types of new technologies. So excited. That's where it's really come out of, is working with universities, as well as a lot of novel late stage discovery assets. So what am I talking about with block phase tomography? And I'll really be focused on cryo-fluorescent tomography, or the use of this with fluorescent labeled drugs. The first image on the left is a 3-dimensional image of a cynomolgus head. This is 5 years old. It's one of the first times we tried taking images off the block face and then reconstructing them in 3 dimensions. It was a hack of a cryomacrotome at the time, and it's actually a bit harder than you think. There's more movement. That image itself was 1800 by 1800 by 1800 40-micron isotropic voxels. That's part of the reason we were able to do it, was we just had the computational power. That's a lot of computational power with a red, green, and blue channel. And in this case, not shown here, there was a radionuclide drug on board that we were looking at the distribution of. That demands 50, 60 gigs of RAM, and it's only been in the last 5 years where that's something that's kind of available. As we move on down the line, I'm going to kind of take you through the most common application of the technology, which is whole whole rodent imaging. There's a highlighted zoom of a brain that's going to be in the oligo application, and then there's a really cool mouse kidney highlighting the distribution of a drug at a really, really high resolution. So this is a macro, a supercellular modality. I'll kind of take you through, zoom into the first plot we had and talk about where this fits. We've used it for just the bright field, just the red, green, blue. We've used it for radioisotope imaging, but its primary application is in fluorescent imaging, and I'm going to show you how that works. After a lot of work on this technology, we launched Emit Imaging, and a great team built the Xerra platform that came out a couple of years ago, and is now kind of gaining traction in the field. I think you'll see why from an imaging and kind of research environment, how powerful it can be. We say CFT is Cryo-Fluorescence Tomography. This system is rather large. The team built a totally brand new cryo-macrotome with a sectioning system that goes somewhere, typically that goes down to 10 microns. We section somewhere between 10 and 100 microns. There's a spectrum of lasers. You can really just kind of image any fluorophore on this system. And as a technology, in vitro, from where this spun out, and now Emit leading its effort, there's been probably 100 research studies run, 100 different compounds studied in this technology. So how does it work? First, you block your samples. We're going to show you different examples of different fluorescent applications one can study. So then you mount this system. You mount the block in the system, and it's really a shaving. We image, we're talking very thin sectioning, and you image at whatever wavelengths you see fit, and then reconstruct a three-dimensional image. It's at face value, the devil is in the detail of the precision of the technology and the reconstruction of the images, and the data I think you're going to find are striking. So where does this fit? I think back to kind of that first plot where we look at the continuous spectrum, there's a big gap in fluorescence imaging, and that's between microscopy and in vivo fluorescence. So this is a schematic. This is a plot that we published. Richard Hargreaves was the first author, a review article we wrote looking at all the modalities and where they fit in terms of, volumetric resolution versus what's the mass of tissue interrogated. And you can see the microscopy, all the different microscopy technologies in the left, and there's a gap, and up and to the right are the in vivo fluorescence modalities. And we're going to show you today that bridging that gap is really this three-dimensional cryofluorescence tomography effort, which interrogates a lot of mass at very high resolution, which is, of course, kind of the key and the future. The excitement of three-dimensional pathology is really about a lot more data and a lot more data interrogated, which we expect and have seen will drive a better AUC for your ROC. So here is an example. We've shown some images, and just for context, I'm going to give a couple ONC examples. This is looking at, on the left, what is standard bioluminescence imaging. Surface fluorescence images don't look particularly, are in the same vein in terms of appearance. And in this case, we have a cell line that could also do fluorescence. So instead of just a luciferin interaction on the left, we also had a cell line expressing. You can see the dramatic difference. Now, of course, the image on the left is an in vivo image, and the image on the right is an ex vivo process. But you do have to ask yourself how much more information content we're seeing on the right, and how much we miss on the image on the left. It's striking. Here's now in vivo fluorescence for a cell line, an ovarian cancer cell line that is throughout the GI, and we also are seeing a HER2-specific fluorophore labeled protein. So you're looking at where did your drug go, and where are the cells proliferating, and just again striking information gain between what is the standard fluorescence technology on the right and what can be done on the left with the Cryo-Fluorescence Tomography. This is a technique we like to call kind of pharmaco-expression. I don't know how real that word is. I first heard it from a colleague, Dr. Jack, and I love it. This is now an AAV9 of which we have transfected the GFP codons in this AAV9. There's control animal on the right. You can see, porphyrin prevalence in the gut, much lighter. Following systemic administration of these AAV9s, a few weeks out, you're looking at transduction of these AAV9s and subsequent expression of GFP. I want to remind people, as I said, I'm a mathematician, so all of this biology typically amazes me, and if I misspeak in any way, I don't proclaim to be a geneticist or an expert in AAVs, that's for sure. But you can see an incredibly striking distribution now of where GFP is expressing. You see the sciatica, the dorsal root ganglia, obviously a lot of hepatic uptake, and just co-registered with a three-dimensional, probably the best three-dimensional anatomical imaging technique I've ever seen. There's no really question of what are the what anatomy you're actually studying. This is just quickly, we get a lot of value from the anatomy. This is the same kidney. I showed you one image earlier. In this case, there were two drugs administered simultaneously, and we can see different distributions in the kidney. And we can actually use the bright field RGB image to segment and pop the vascular of this kidney. So really, really cool. And I want to give a shout out to Rob Holt for many reasons. He was one of the primary drivers of this technique, and he was never credited enough with that awesome review plot earlier. A great optical scientist who did a lot of this work. And now to another ONC experiment. This is work from Hemi DeMont from Vicro, also Eric Ahrens at UCSD, who uses the system and developed a very, very nice nanoparticle construct that has embedded a fluorescent compound embedded into it. That's the red channel you're seeing here. You're looking at really tumor-activated macrophage and other macrophage imaging across the whole mouse, and in conjunction, the GFP cell line, and you're also seeing gut from autofluorescence. Co-registered now, you have really the two images co-localizing the distribution of this V-Sense macrophage imaging agent, as well as the cellular proliferation of the GFP cell line. So it's super useful in a lot of modalities, and that kind of sets the stage now for a Society for Neuroscience killer application. So much credit to I don't think I've ever put just a research article to this state in a page, but I'm really going to dive deep into the work of Kurt Mazer. I'm a co-author on this paper, but many people contributed to what is a very multimodal, multi-omic activity, and it looks at what are the pharmacokinetics, the biodistribution of the antisense oligonucleotides. This work done by Biogen and Ionis in partnership with the work was done at Vicro with the Emit platform. And so let's talk out the gate. So this is about a rat study with intrathecally administered ASOs. There's a control gene, MALAT1 and GABA1, modifying GABA expression. So there's two oligos, two cohorts. The first study was iodinating them and imaging them in vivo over time, over 7 days. And you really get to see a fantastic, what I talked about, microliter biodistribution of where your drug's going over the course of a week. It's a phenomenal image. It tells you a lot about clearance, PK, but it's not going to tell you about, a lot about what's happening in an organ, or certainly not what's happening at the cellular level. So I'm going to take you through the next studies as well as, in this case, a pharmacodynamic study. So out the gate, in this case, brains were resected. We have the anatomical images. They're superimposed here with the fluorescence. In this case, there's 4 different rats, one sacrificed at each of these time points. We have more time points showing. And that really is insightful. We're really seeing kind of the distribution of the oligo in the brain over time. Here's now in 3 dimensions, that image. We're looking at, this is 6 rats over 12 hours. We're kind of seeing the wash in and then distribution scaled quantitatively of where is the oligo moving. Because in this case, we then co-registered all of these brains to a rodent brain atlas and looked at per region of brain, what was the distribution over time, and very insightful as compared to where things are going in the cortex versus right. I'm not a neuroscientist, I apologize. So my brain anatomy is not that great, but have done a lot of rat brain imaging. And here you see now a very clear distribution schedule and very insightful as to kind of where oligo is going in the rat brain. Now let's go down another 3 orders of magnitude in volumetric resolution. In this case, the team did a phenomenal job. You can stop the imaging, remove a section, and look at it, and do whatever histopathology you'd like. And in this case, you have the fluorescent oligo on board. So here's an array of different stainings, as well as an understanding of where was the systemically administered, so co-localized with these other cells. The imaging scientist in my case is you've covered now from femtoliter, from the subcellular all the way through 4-dimensional distribution of the drug. You have incredible control over where the drug is as a function of time and location in this model for both cohorts. Now, PK is actually already a huge win to successfully understand this level of PK from cell to whole organism. But a next step is what's happening from a PD perspective. So what is the drug doing? In this case, we look at it's a GABA. It's knocking down GABA expression. That's what we was expected this oligonucleotide was doing. So we use flumazenol to measure GABA expression in these brains, and we see consistency across the MALAT1 gene, the housekeeping gene, and then for the GABRA1 ASO, we see a knockdown over time. This image on the right now is taking these flumazenol scans. Each row, this is 4 rats co-registered, 4 rat brains co-registered at 4 weeks for the MALAT1, for the GABRA-A, and now we're looking at a voxel-wise area under the curve difference. So this is the area under the curve for flumazenol, average across 4 rats on the top, the middle, and then the voxel-wise AUC comparison of the 2 cohorts, and it's obviously statistically significant and a clear-cut drug effect. So, and having run a lot of these studies, I think the value proposition here is we often have to run many, many studies to paint a picture of the pharmacokinetics and pharmacodynamics of a drug. This is a single study. I wouldn't say it's not a complicated study. I wouldn't say it was an inexpensive study. But if you think about how many things it answered, price per data, this is a powerful technology deployed to great effect, which is why I have a bias towards that technology. So again, I think whatever your basic science needs are, or I just think there's a lot of value in using this block-based technology technique. Currently, the form is really on a bigger block. It's a cryo-macrotome technology. The company is evolving it for more microscopic endeavors. But as it stands, another great technique, and this one was done with the radionuclide. People can and I've started using it for resected fluorescent organs. to do a block, you can think of a block as a day's work. And here now in a day's work, there's 80 rodent brains in a block. And so there's actually 10 cohorts in this case. And we've sectioned, in this case, we transferred the tape, the team, credit to Invicro and a collaborator, transferred via tape to a phosphorimager and was able to reconstruct in 3D the distribution of in this case, a C14 drug. This is actually quite a lot of work with a radionuclide, but it's a lot less work with a fluorophore. You can get now one block, you can have as many samples as you fit in that block. This study is very cool, very impressive what they did. They then took each brain and co-registered them to a common space and looked at a voxel-wise comparison of the distribution of the drugs across the different cohorts. But a lot of data, so resolution and sensitivity are always a trade-off, but there's a lot you can do with this technology across a lot of applications. So thank you for your time. I want to acknowledge the Emit team. I want to acknowledge Jacob Hesterman and the entire team at Invicro. So many people put so much work into this. AJ Verma, Kurt Mazer, a lot of people at Biogen, specifically on that deep dive application. I want to thank the people that contributed to that publication, of which I was an honor, and many others. It's been a lot of work. And so thank you for your time, and don't hesitate to reach out with questions. Thank you.